Hello, my name is Deontray. I will be discussing the gut microbiome project that we went over in class. I will start by discussing what a microbiome is and what gut microbiome is. So the microbiome is the makeup and distribution of microorganisms within a certain environment, and the gut microbiome is the makeup and distribution of microorganisms pertaining to the digestive tract of a animal. So the reason that we are interested in the gut microbiome is because it has been linked with many different physical and mental conditions, health conditions. And so, for example, we have a study that looks at CDI, which is Clostridium difficile infection. So basically, Clostridium difficile is a bacterium that, after a successful antibiotic treatment, which can wipe out the majority of a person's normal gut microbiome, will come in and act as a founder species. The problem here is that this bacterium produces harmful endotoxins, and through preemptive competition, outcompetes most other bacteria that try to colonize in the digestive tract. The study conducted took patients known to have Clostridium difficile infection, CDI, and treated them with an antibody that would target the class of family that Clostridium difficile resides in, wiping out the bacteria. Then, after performing a fecal matter transplant from a healthy individual into a previously infected individual, researchers observed increases in healthy microbiota and a decrease in harmful microbiota, as well as a decrease in further CDI infections or Clostridium difficile infections. This suggests that there is a correlational link between Clostridium difficile infections and the gut microbiome, and that Clostridium difficile infections may be treated through microbiota-based therapies. And then another study we have that linking the gut microbiome to depression. Or this one, while the first study was causal, this one is more correlational, because it links the gut microbiome to mental health labeling relationships, as recent studies have linked it to the to mental health labeling it as the gut-brain axis. And this study shows a correlation relationship between the taxa of microbiota found in the digestive tract and the presence of depression in a patient. The experiment identifies and compares the taxas of microbiota between two groups, those with and without depression. The largest number of differentiating taxa here was within the Firmicutes, in which 9 families and 12 genera differentiated the group with depression. The majority of these families and genres were found to be statistically different between the two groups in the two identified studies. So those are these studies just go to show why we think why we're interested in the gut microbiome. And so this going into our the actual experiment that we performed is here goes our experimental question or hypothesis. So we hypothesized that the taxa of the gut microbiome between 2 guinea pigs will be the same because of them being the same species group, and will differ across species with a iguana. So basically, going up here, this shows this table here shows the animals and backgrounds of the animals that we use for our specimens to extract fecal matter for our gut microbiome analysis. You can see here that we use 2 similar conditioned guinea pigs. See, we have pretty much 2 similar conditioned guinea pigs, and that we compare. After comparing microbiotas, we expect them to be highly similar to one another. And then we have iguana, which is an entirely different species, and we expect that to be highly different from these 2 species and to have its own makeup of its gut microbiome. So then, going into our methods, basically, we have 3 main steps. So for our action experiment, we have 3 major steps, which are DNA isolation, polymerase chain reaction, and nucleotide sequencing. Our first step, DNA isolation. This starts here. Our first step, DNA isolation, consists of 2 processes: the Quadrant kit and the Zymo kit. The Quadrant kit lyses cells through the centrifugation and homogenizes the stool in a special buffer that prevents enzymes from degrading the DNA. Then the DNA is bound to the filter wash of impurities and eluted from the filter by a buffer that weakens the bonds between the DNA and filter. The second process, the Zymo kit, binds the the purified DNA to a zymo spin column and then washes it again before being eluted by the filter from the filter by a special buffer at 1/10 the initial purification, causing the DNA to be 10 times more concentrated. So again, this one basically isolates that DNA, gets the DNA out of the cell, and this in the zymo kit makes the DNA more concentrated. So then as we go on to our next step, we have PCR Yes, so the second major step is PCR. It consists of 6 major steps: DNA quantification through spectrophotometry, PCR or polymerase chain reaction, gel electrophoresis, PCR cleanup, indexing PCR, and indexing PCR cleanup. Starting with DNA quantification, which would be here, we're looking at our DNA quantification. So we calculated the concentration of the DNA by reading the absorbances of 260 nanometers and checking for proteins or RNA contamination by using the ratio 260 over 280 nanometers. And so that's basically just showed us that our DNA sample is pretty pure. It doesn't have any contaminants inside of it, and that it was concentrated. So then we go into amplification of our DNA by PCR. We wanted to amplify the 16S ribosomal RNA gene, a gene with variable regions interfering between the conserved regions. Conserved regions allow us to identify the gene, and variable regions are used to identify bacterial species. We start off the PCR process by adding 2x master mix to our DNA solution. Then we place the solution in a thermocycler at temperature of 95 degrees Celsius, which promotes the DNA denaturation, separating the 2 strands. The thermocycler then switches to 55 degrees Celsius to promote primary annealing. Lastly, the thermocycler changes over to 72 degrees Celsius, allowing for the DNA replication by polymerase. After many cycles, the DNA is amplified into the millions. So we just want this specific gene of DNA, the 16S ribosomal region, we want many copies of it so we can use for our samples, so we can sequence it. So going on to our next step, which is gel electrophoresis. So for gel electrophoresis, we are measuring to see if the PCR amplification was successful. We place the DNA into wells in a agarose gel between the 2 oppositely charged poles. The DNA is placed on the negatively charged pole anode, and it ends up migrating to the positively charged cathode, and due to its negatively charged phosphate backbone. Distance is moved is inversely proportional to size, and because we have known DNA markers of the 16S ribosomal RNA gene, we can observe if the DNA in our sample lands on that marker to know if it is the right gene that was amplified. So then, again, we're just checking to make sure our PCR work. The process. So after gel electrophoresis, we do PCR cleanup, where we use the Angicor Ampure Kit XP kit to remove compounds of 2x master mix. This is done by binding the DNA to metal beads, washing it of impurities, and then eluting and collecting it in a new buffer solution. So that's just, again, washing the impurities. The next process to be performed is the indexing PCR. Because all student samples will be mixed for DNA sequencing, there needs to be a way to distinguish our DNA from the rest. This is done by adding unique nucleotide sequence tags at the 5' end of our DNA through a special PCR. So that's just, again, we are going to have to end up mixing all our samples, so we need a way to identify it, and that's for our indexing tags. The last major step is to sequence our DNA, and to do this we use using fluorometric DNA quantification and aluminum MESEC next-gen sequencing DNA analyzer. The fluorometric DNA quantification uses a Quibat fluorometer to measure the concentration of the amplified DNA, making it possible to determine the correct dilutions for the sequencing reaction. And the aluminum MESEC next-gen DNA analyzer uses a flashlight to record at what length each nucleotide absorbs that, and therefore determine the nucleotide sequences of the DNA. The DNA sequence was compared to a database, and that database, and because of the variable regions of the 16S ribosome RNA gene, the database was able to distinguish what species of bacteria the DNA actually came from. So then if we go up to here, we're actually going into start looking at our results. So we're going to see that we have, this is our results for the quantification of our DNA, and our quantification of DNA step. And so basically we're seeing that we didn't really have any contamination, and that our concentrations were correct. And then this right here, one down here, just shows that our electrophoresis. It just shows our positive, control, negative control, and our different samples. And if we look at the bands here, we can see that they line up with our known and expected bands, meaning that our amplification process, or PCR, actually worked. And so then going down to our fluorimetry, we see that we have some of our samples actually didn't pass this step, so we didn't get results for them just because we must have had some problems go wrong. And so, but we did get some samples to actually run through, and so this shows shows us where the concentration of our DNAs at this point in the step in the process. And so then to go into our last one, our reads, this shows how the guinea pig DNA, and after we got our reads for the guinea pigs and the iguanas. And so